It's a real pleasure to introduce Lucy Davison in the second talk. Lucy is a MRC clinician scientist working at the Royal Veterinary College, a specialist in small animal internal medicine and a professor in clinical genetics at the Royal Veterinary College. And she is going to talk about some elements of her research here, looking at the genetic and environmental basis of complex diseases across a number of species. As you can see, Lucy is going to be talking about canine respiratory coronaviruses here and the role of canine of host genetics. Lucy, thank you very much, and over to you.
Great, thank you very much. Hope everybody can hear me okay. Yes, as James has very kindly introduced, it's a real pleasure to be here talking outside my comfort zone on virology. I'm a vet by training. My background's in diabetes research and endocrinology, but as with many of us, the pandemic drew us into viruses as well. I want to start by acknowledging a range of different people who've been involved in this project, particularly Judy Mitchell, who is the expert on canine respiratory coronavirus. She's worked in it long before it became much more trendy, and she's been extremely generous and helpful and knowledgeable in this whole process. Tom and Sarah and Marsha have also done the bulk of the work that I'm going to present here today. And this is all really as the result of a UKRI grant that we got early in the pandemic, looking at susceptibility to coronaviruses in dogs and cats, and all of the members of that consortium are shown on this screen. So today I'm going to talk mostly about canine respiratory coronavirus. I'm talk a little bit about the clinical presentation. I'm going to talk about our mascot project, looking at host genetic susceptibility, the transcriptomic landscape of respiratory coronavirus in the host, and our attempts to try to identify viruses in infected tissues. I will give you a little bit of a warning that that is not as exciting as you might hope, so please don't get your hopes up for that section, and a little bit on future perspectives as well, and I'm very happy to answer questions at the end. And we've been very fortunate to have a great talk prior to this one, so I don't need to do too much introduction. But what we're talking about in this particular presentation is a beta coronavirus, which is canine respiratory coronavirus. And what this is a disease that affects many, many dogs. It's highly contagious. And it has been reported to have quite a lot of overlap in interest with SARS-CoV-2. If you want to learn more about that, there was this commentary written actually in July 2020 by one of my colleagues at the RVC, and I would refer you to that. But just to show you how it relates to its closest neighbours, there is quite a lot of relationship between canine respiratory coronavirus and also the bovine coronavirus, and one particular human coronavirus. This is a paper published in 2019. Very much more recently, in fact, this year, there has been a much more comprehensive comparison of canine respiratory coronaviruses in the US and the closest related beta coronaviruses from other species, and also the human coronavirus OC43, which is the closest to the canine respiratory coronavirus in sequence. This is a summary, and as you can see, all the different isolates from dogs in different countries are listed down the left-hand side here. And then we have bovine coronaviruses and the human coronavirus at the bottom. So you can see the level of identity that we're talking about. There's still quite a few mysteries in canine respiratory coronavirus, and this is an area that Judy Mitchell and her group are very actively working on. Clearly, these related viruses all differ in species specificity, and that's a really important and interesting observation. They all bind to sialic acids. That appears to be permissive for the human coronavirus, and it seems to be attachment only for the canine and bovine coronaviruses. There may also be caveolin-dependent endocytosis contributing to entry, and there is some discussion too about MHC class 1. So when we look at exactly what this virus does and how it manifests, it was first discovered in the UK in 2003, and that was associated with an outbreak of severe respiratory disease in a canine kennel rehoming setting. Obviously, it's not the same as canine enteric coronavirus, as we've just heard. It's very rarely that I have the opportunity to present after somebody who's talked about the enteric coronavirus. So it's great that we've already discussed that. It's present worldwide, with 50% of dogs in the USA and Canada having antibodies. We've already said it's a beta coronavirus, and there are some really important areas of identity between the antigenic epitopes of the canine respiratory coronavirus spike protein and SARS-CoV-2. It's extremely contagious. If a dog who's previously unexposed goes into a shelter setting, whether that's rehoming or just a boarding kennel, it will seroconvert within 3 weeks. And that's where the name kennel cough has really arisen, because this really affects mostly dogs who are closely confined and sharing airspace. One of the other similarities that really made us most interested in this virus is the fact that there's a spectrum of severity. Some dogs get it and they're completely asymptomatic and they seroconvert. Other dogs succumb to pneumonia and may even need to be euthanized because they get such severe respiratory signs. And we know that's not just a virus. We know that there are other pathogens involved in that, but even within the same setting, with the same pathogens in circulation, some dogs do really well, other dogs do really badly. And this so-called canine infectious respiratory disease complex is one really good example of multi-infections contributing to different outcomes. We don't use antivirals for these dogs. There isn't a vaccine, but if possible, not always feasible in a kennel setting, but if possible, dogs with kennel cough need to be isolated from other dogs for 3 weeks, and we provide them with supportive care. When we look at what is behind the canine infectious respiratory disease complex, there are many other viruses, but there are also Bordetella, Mycoplasma, and Strep that can be involved in the presentation of kennel cough in dogs as well. This paper published last year shows a relative proportion of different organisms and different things isolated from dogs with respiratory disease complex. You can see canine respiratory coronavirus is in there, but there is also quite a significant proportion of Mycoplasma in there. What is interesting, though, is if a dog has neutralizing antibodies to canine respiratory coronavirus, when it arrives in a shelter, it is far, far less likely to develop canine infectious respiratory disease complex or kennel cough. So clearly this is a virus that's important in a critical early stage. Now, Simon Priestnall and Judy Mitchell and Joe Brownlee back in the early 2000s really started to work on this virus and have done a lot of work looking at its impact on different cells and the pathology of infected dogs. What we see in the trachea is clumping of the cilia and necrosis of epithelial cells and a lymphocytic, plasmacytic infiltrate. And we can also see that the epithelial and goblet cells become virus positive. And what happens in the dog itself is the dog will develop a dry cough, sometimes a nasal discharge. If we look at them hematologically, they'll have a lymphocytosis after about a week. They may also have a virus-associated drop in neutrophils, so neutropenia. And experimentally, virus has been detected not just in respiratory tissues, but also actually in spleen and lymph nodes and the GI tract. And it's hypothesized that the way in which canine respiratory coronavirus predisposes to canine respiratory disease more complex is that it damages the mucociliary clearance mechanisms, and so you get predisposition to other infections. So the canine respiratory virus infection usually occurs pretty quickly in a shelter. If you're going to get parainfluenza, that tends to happen later, and canine herpes is even longer. And the limited studies that have been done so far suggest that in the early stages of infection, the virus may inhibit pro-inflammatory cytokines and chemokines. After about 1 or 2 days, drops in IL-6, IL-8 and TNF-alpha. So that's the background. Looking at what we've done with the Mascot project, as I said, my background is in endocrinology. I'm an internal medicine specialist, so I came to this during the pandemic with the idea that we might be able to adapt some of our work studying the genetics and host susceptibility, as it were, to canine diabetes, looking across to coronaviruses. Diabetes is a disease which we know is a combination of genes and environment, as is FIP, as is canine respiratory infectious disease complex. So I think we have quite a few parallels there in that we're modeling not only host genetics, but also a fairly stochastic environmental event as well. We know what that environmental event is in the case of viruses. It's a bit more mysterious in diabetes. So this is the slide really that we use to try to secure the funding, and that we use to outline the project from the canine and feline point of view when we're presenting it to others. And as I've already mentioned, this beta coronavirus is highly contagious. But the critical point here is that because of the work done in the early 2000s by Joe Brownlee's group at the RVC, there was already in existence a large frozen tissue sample archive. And that is what has underpinned all of this research, really the foresight of those people involved at that point, banking those samples so that we could return to them promptly during the pandemic. And we know that most dogs recover quickly, but some dogs develop fatal pneumonia. And the idea being here that we do whole genome sequencing and MHC analysis, comparing severely and mildly affected dogs, following that up with targeted sequencing to try to find candidate variants that in the host determine how well a patient does. And then to inform some of that decision making about which genes we would focus on for our genomics of the host, we also did some RNA sequencing of canine respiratory virus affected and unaffected lungs. And we had those samples, as I said, already archived, because these dogs, some of them had been euthanized for their kennel cough, others had been euthanized for other reasons. So we had a spectrum of different lung samples to choose from. The idea being that some of these outcomes would be translatable to COVID-19, looking for genetic susceptibility to severe disease, maybe finding coronavirus treatment targets, understanding companion animal responses to coronavirus, and thinking about future pandemics and maybe zoonoses. So I'm going to briefly now talk about the host genetic susceptibility work. And as I said, the lung tissue samples were collected post-mortem at a single shelter from dogs euthanized for canine infectious respiratory disease and other causes, and those had been archived in frozen storage -80 at the RVC. And this picture here shows the 4 different categories, 6 dogs per group that were examined in this really pilot work. It was our first foray into virology. We have dogs who are negative for coronavirus, and they're shown in red and green, but some of those dogs had no lung pathology at all, and others had quite severe lung pathology, but no evidence on PCR of coronavirus. Meanwhile, the coronavirus positive dogs either had no lung pathology in this teal colour, or they did have very severe lung pathology in this purple colour. And the plan was to do RNA sequencing of these dogs, whole genome sequencing of these dogs, and then follow up with the remaining dogs in the archive, the genotyping to see if we could find host susceptibility factors. What you will notice if you look at any studies from a shelter is that many dogs are male, most of them are very young, and that is just a fact of life. That is the dogs that end up in shelters. Also, as you'll see here, despite our attempts to breed match, many of the dogs were crossbreed. We had a few Staffies, German Shepherds, Lurchers and Rottweilers, but the majority of dogs here were crossbreeds. Now, before I talk about the data, it's worth mentioning that we learned during our time studying canine diabetes that we have to be careful about how we classify genetic variants in inbred populations like dogs, because of the population structure and the way the different breeds are actually quite distinct and have quite limited genetic variability. But then between breeds, we have quite a lot of variability. And sometimes in the case, for example, of diabetes, we find whole breeds that are predisposed to the disease. So actually the variants we're interested in are exclusive or distinct to that breed. And that's what we call class 1 variants. They're high frequency and fixed in a high risk breed. It's kind of quite hard to know what a high risk breed is in this situation, but we have to be mindful that there may be some fixed variants. And then there's also the variants, if you were to take a whole breed and look at cases and controls within a breed, variants that impact on susceptibility to the condition within the breed. So they're seen in more cases than controls. And then there's a few other strange, unique variants that might go and affect individuals. So we have to be mindful of this population structure. And that was helpful in adapting our bioinformatic pipelines to understand what was going on in canine respiratory coronavirus, having previously designed it to look at diabetes. So I'm not going to go into the bioinformatics in a lot of detail, but the pipeline for our whole genome sequencing is pretty standard, using GATK as our variant calling and joint genotyping process. And what we've done is we've developed a system of variant prioritization from the many millions of variants that we identify from whole genome sequencing. So in these 24 dogs alone, there were 13 million variants to prioritize. We wanted to try and pick our top 5,000 that we thought might be impacting on disease. And to do that, there's a range of different things that go into what we call an evidence score. And that might be whether the variant's been associated with other the gene has been associated with other viral infections, what the predicted impact of that variant is, and whether the gene has previously been associated with immune responses or viral diseases in other species. So as well as looking at the alternate allele frequencies and the actual the genotype and the relationship between cases and controls, we also try to prioritize by variants that are in genes of interest. We, of course, also feed our RNA sequencing data into that as well, because that also helps us to be a little bit more focused on the specific disease. So the other thing that we do is, as well as taking our 12 cases, 12 controls, coronavirus positive and negative, we've actually got a load of other whole genome sequencing data from other projects in the lab. So we use that to integrate into the data set and try to understand whether some of the variants that are segregating with poor or good outcomes actually are common in the population or not. And the upshot is that we end up with the capacity to segregate variants according to which group they are in. So this is a heat map, and it's just showing, for example, the variants where the severe dogs, so those are those dogs with virus positive and really bad pathology, where the vast majority of dogs have 2 copies of the allele. So this is shown in yellow here. It's a heat map. So if they've got a high frequency of this particular variant, it's in yellow. The darker the color, the lower the allele frequency. We've also got access to a range of other databases from the Animal Health Trust, from the Dog Biomedical Variant database, and the Dog Genome Project, which tell us about the background level of that allele in the population. And then we've got our other groups here, our virus negative groups and pathology positive and negative groups. And you can see here that we've pulled out the variants that are really strikingly at higher frequency in those dogs with really severe coronavirus and pathology. And that they are ones that we'll prioritize for going into follow-up genotyping. As I said, I'm not going to show you every single thing that we've done, but essentially we've identified 5,000 variants that we want to take forward, or we are taking forward for further sequencing. And you can see on this particular first graph, the evidence score, we have a lot that are, they're reasonably associated, but we're really interested in these with the highest evidence scores to take forward. We filter those, and we've ended up with a number of targets, that some of which are from our RNA sequencing, differentially expressed genes, some of which are high impact, and some of which actually are boosted with a higher evidence score because we know they've previously been associated with infection or immunity in other studies. And this is an example of some different evidence scores and the types of evidence that the boxes that are ticked by each variant. So this is just a handful of variants, a different variant on each row, and you can see the different evidence scores that have been added up computationally. And you can see here that, some of these are genes that have been associated with COVID-19 severity. Some of these are genes that we know are expressed in the immune system. And we will basically add up these sources of evidence, and that will be how we pick our top 5,000 which have gone forward. I just want to show you an example of how this plays out in reality with some individual variants. So these are 3 variants here in genes, separate genes involved in the innate immune response. And you can see here we've got the population allele frequency is on the left-hand side, and you can see that the homozygous reference dogs are in green, the heterozygotes are in blue, and the homozygous alternates are in red. And what you can see is the Animal Health Trust, the Dog Biomedical Variant Database, and Dog Genome Project allele frequencies are shown in their own specific columns. And the dogs I think we're most interested in are those dogs who have coronavirus. So this is column 4, the dogs who have coronavirus but don't seem to be having a really bad time with it, and the dogs who have coronavirus and doing really badly. That's the interesting comparison. And you can see here, just for 3 selected genes in the innate immune pathway, we've got some variants, for example, where all the dogs who've got bad pathology and coronavirus are homozygous for this variant. So we think that may be predisposing them to bad outcomes. Similarly, we've got some variants where the dogs who have, as in the first block, we have some dogs who don't have any pathology, but you can see they're coronavirus positive, but none of them are homozygous for this alternate allele. So it might be that that's also providing some protection. What we're doing is we've now just received the data back from 100 dogs. We've genotyped them at 5,000 prioritized variants, and we look forward to sharing those results with you. The first look is pretty compelling, actually, and I think we're going to learn a lot, not just about coronavirus, but about differences in dog immune systems by breed, and also by innate immunity variation. The host transcriptomic landscape is also something that's fed into this project. Again, I'm showing you the four groups here, and the dogs that are teal and purple as before are those with coronavirus. The teal have no pathology, the purple have really bad pathology. And I think on this global transcriptomic picture from the lungs, you can sort of get a sense that these teal dogs are segregating away, and we thought this is quite interesting. Maybe there's something going on in the dogs who have coronavirus but no bad pathology that is somehow protecting them. And when we look at the heat map, which shows us the gene expression signatures, you can see the dogs are clustered here again by bar at the top of the heat map. We have the coronavirus negative, the coronavirus negative and positive, negative dogs here on the left, the coronavirus positive dogs here on the right. And what you can see in the first block, cluster 2, are the genes that are expressed in dogs who have coronavirus but no pathology. And cluster 1 is the genes that are expressed in dogs who have coronavirus. They're differentially expressed in these dogs who have coronavirus and bad pathology. But do note that there's quite a lot of individual variation here. There isn't a single signature, I would say. So what do we see? Well, it turns out that cluster 2, which is the coronavirus positive lung pathology negative dogs, this is on the right-hand side, the biological pathways that are really represented with the differentially expressed genes. And they seem to be about metabolism, and particular immune cell metabolism. There is a real difference in the immune cell metabolism of those dogs who have coronavirus but don't seem to be having a major problem with it. So that's really fascinating and something we're keen to explore further. Cluster 2, as you might expect, the dogs having a bad time have a really, really bad inflammatory response. And we can break that down into looking at individual genes. And I'm not going to expect you to look at every single gene here, but what I would say is many of the differentially expressed genes, particularly between the dogs having a good time and a bad time with coronavirus, are along the lines of the TLR genes and cytokine genes and cytokine receptors. You can see quite a dramatic difference between the teal and the purple in many of these genes, according to whether or not they are having a severe response to coronavirus. So I think it does show that even though these samples are 20 years old, we've still been able to get some interesting data from them. And I think that is a really good model of spontaneous respiratory coronavirus infection. And we've shown the cytokine production, cell chemotaxis, inflammatory pathways are critically important here. And we've also shown this metabolic dysregulation or metabolic change that seems to distinguish those dogs who react to the coronavirus and who don't. So finally, I just want to mention our attempts to try to pull out viral reads in the canine lung sample. I hope I tempered your enthusiasm for this early on. These samples have not been stored in RNA later. We've had much, much more success with feline tissues for FIP stored in RNA later than we have with these snap frozen canine lungs. So again, these 4 different categories, what we have done is listed them here consecutively. So we've got the viral negative samples at the top. There's a block of 6, another block of 6, and the virus positive samples here at the bottom, a block of 6 and a second block of 6. And in the first block, we have those who don't have really bad lung signs, and the second block is those who have really bad lung signs. So actually, we got a few surprises here, and I think this is really because we are not powered to see a great deal of viral transcript. I think a lot of them have degraded. One thing we did notice, I suppose it's reassuring that our most successful ability to pick up viral transcripts was in the dogs who had virus and really bad lung signs. But we did have, when we mapped what we found to lots of different viruses, different canine respiratory virus isolates, but also bovine coronavirus isolates, we had this mysterious mapping of one set of to one particular canine respiratory coronavirus isolate, even in those dogs that have negative by qPCR, which seemed a bit strange to us. And what actually we found there was that, in fact, that particular full-length canine respiratory coronavirus genome sequence actually does contain some canine mammalian sequence, and I think that's what we were mapping to in the end. So we were slightly relieved to discover that our negative dogs really still genuinely are negative. We just don't have a great deal of information on our positive dogs, I think due to degradation with time. So as I said, the coronavirus sequence being detected in all samples just for that one isolate was likely due to sequence contamination from mammalian sequence. And let's go back and look at bovine. Again, we have limited capacity to really detect bovine coronavirus. We're not quite sure. We need to go back and look at this in a bit more detail. We need to look in more detail at the actual sequences we've been able to detect, but extremely patchy, much, much more successful, as I said, with RNA later samples in the felines. So just to finish off, what we're doing next, we're awaiting our targeted genotyping results. We've got the data, we're just analysing it. We're still doing MHC genetics, although I think we're a bit underpowered with our initial cohort. We're waiting for the large cohort DNA samples to be able to do that. We're looking more closely at the viral reads, and we've also got data on the multiple pathogens in these dogs, and I think that might well be important as we come to segregate them. We're quite interested in combining serological and genetic data. We haven't done that yet, but we do have serum samples archived from some of these dogs. And of course, we're interested to see whether our variants that are predisposed to canine respiratory coronavirus are underlying SARS-CoV-2 susceptibility in dogs. And there are definitely going to be collaborative opportunities now to think about a vaccine for this disease in dogs, because I think its primary role in kennel cough probably makes it a very good target. My final slide is some unanswered questions, just in case you were thinking of asking me. We don't know how long the immunity lasts. I don't really know whether a vaccine is required, but if one could be made simply, I think it would be very valuable. We don't exactly know which breeds are susceptible and why, because of the mix of dogs that end up in shelters, it's not representative of the general population. Nobody, as far as I know, has used antivirals in kennel cough. We may be able to design other immunological interventions, and I think actually the canine respiratory coronavirus might be a good arena in which to explore that, because I do think genuinely this could be an appropriate spontaneous model for SARS-CoV-2 studies. So that's the end of my presentation. I just want to thank everybody again. I've put my 2 email addresses there. I answer both equally. They're one Oxford, one RVC, and I'm very willing to have questions by email, but also to answer questions now. So thank you very much for your attention.